by starting with the digital code in the computer, building the chromosome from 4 bottles of chemicals, assembling that chromosome in yeast, transplanting it into a recipient bacterial cell, and transforming that cell into a new bacterial species. So this is the first self-replicating species that we've had on the planet whose parent is a computer. It also is the first species to have its own website encoded in its genetic code. But we'll talk more about the watermarks in a minute. This is a project that had its inception 15 years ago when our team, we called the Institute Tiger, were involved in sequencing the first 2 genomes in history. So it's been a long route to get here, 15 years from the beginning. We felt one of the tenets of this field was to make absolutely certain we could distinguish synthetic DNA from natural DNA. Early on, when you're working in a new area of science, you have to think about all the pitfalls and things that could lead you to believe that you had done something when you hadn't, and even worse, leading others to believe it. So we thought the worst problem would be a single molecule contamination of the native chromosome, leading us to believe that we actually had created a synthetic cell when it would have been just a contaminant. So early on, we developed the notion of putting in watermarks in the DNA to absolutely make clear that the DNA was synthetic. So the team actually developed a new code within the code within the code. So it's a new code for interpreting and writing messages in DNA. So there's 4 major watermarks, all over 1,000 base pairs of genetic code. The first one actually contains within it this code for interpreting the rest of the genetic code. So in the remaining, we're here today